Chapter 8, Lymphocytes. Lymphocytes are principal effector cells of the adaptive immune response system. What that means is they develop slowly and they generate a response to eliminate pathogens. There are two types that function to eliminate these molecules, and they work in two different ways, either through immunity or cell-mediated immunity. Their primary function is to recognize and react with specific antigens, work with macrophages to eliminate foreign invaders. They provide long-lasting immunity to any previously encountered pathogens, and their lifespan can be several years, or they can die within the circulatory system in hours to days. There are three types of lymphocytes. B lymphocytes that are primary effector cells for the humoral response, which is the production of antibodies, and they must be activated to differentiate into the plasma cell. There are natural killer cells, which are effector cells of innate immunity, and T lymphocytes that are primarily involved in cell-mediated immunity. Figure 8.1 is a great visual demonstration of how the hematopoietic stem cell commits to the common lymphoid progenitor and then matures into the three different lymphocytes, except for the T lymphocyte, which matures in the thymus. Now, during these maturation phases, they're released as immunocompetent lymphocytes. Lymphocytes are unusual because once they become encountered a foreign antigen, they undergo a transformation to become the cell needed for specific immunity, either humoral or cell-mediated. So first we're going to follow the path of the B lymphocyte. So B lymphocytes, we know, are these precursors that arise from the bone marrow from the common myeloid progenitor. They undergo antigen-independent phase of development in the bone marrow, and they exit as naive B lymphocytes. B lymphocytes compose about 15 to 30% of our peripheral blood lymphocytes in circulation. They are identified by the presence of their surface immunoglobulin. And these markers are identified at various stages. During antigen stimulation, some activated B cells undergo isotype switching at the constant region of the heavy chain gene, converting to either IgG, IgA, or IgRE. Stimulation from the mature B cells undergo proliferation, and they differentiate into these antibody-secreting plasma cells, and these plasma cells proliferate into memory cells, which gives us our antibody production. Now let's talk about the T lymphocytes in this chart. The T lymphocytes come from the bone marrow as naive cells, and they mature within the thymus. They are antigen independent at this point in time. Now, with the major histocompatibility complex you learned in immunology, they're involved in recognition and interaction between these T cells and other cells of the body, such as infected host cells, macrophages, B lymphs, and dendritic cells. MHC antigens, called human leukocyte antigens, function to present antigens to these T lymphocytes. They must be recognized by these T cells to elicit a specific immune response. There are two types of T lymphocytes. CD4, which are T4 cells, which are the most prominent type of lymphocytes in our lymph nodes. They take up about 60 to 80% of our peripheral blood lymphocytes. CD8 or T8 cells are predominant lymphocytes in the bone marrow. and take up about 35% of our peripheral blood T lymphocytes. The T4 to T8 or CD4 to CD8 ratio in circulation is 2:1. This is very critical for normal activity of the immune system. When they encounter a specific antigen, they undergo an immunoblast transformation. These become activated lymphocytes and help provide cell-mediated immunity. The third population of cells are natural killer cells. They are effector cells of the innate immune system. They originate in the bone marrow and usually consist about 5 to 15% of lymphocytes in the peripheral blood and the spleen. They have characteristics of both natural killer and T cells to help eliminate foreign invaders. All of these lymphocytes undergo the same maturation stages. It is very hard morphologically to identify the difference between T, B, and natural killer lymphocytes. So there are other tests to determine the type of lymphocyte they are. For you as an entry-level CLS, all you have to identify them as lymphocytes. So for the maturation stages, each cell within the bone marrow undergoes these transformations. Lymphoblast to prolymphocyte to lymphocyte. So let's talk about the first cell of maturation, the lymphoblast. This is a very large cell, 10 to 18 microns. It has a very high nuclear to cytoplasm ratio. The nuclear chromatin is lacy and fine, and it's more smudged than you would see in a myeloblast. They usually have 1 to 2 well-defined pale blue nucleoli, and the nuclear membrane is usually dense, and sometimes you can see that perinuclear clearing zone. The cytoplasm is agranular and usually less basophilic than you would see in other white cell blasts. The prolymphocyte is the next stage in development. It's difficult to distinguish in the normal bone marrow. It is slightly smaller than the lymphoblast, and it has a lower nucleus to cytoplasm ratio. The nuclear chromatin is clumped, and you have finer chromatin than that of a lymphocyte. You can see most of the time one nucleoli present, and the cytoplasm now is starting to take up a lighter blue, but is still agranular at this stage. Now, when we get into mature lymphocyte morphology, because there are different types of lymphocytes, we will talk about the normal types of lymphocytes seen. Small lymphocyte is the most common type. It is 7 to 10 microns in size, and usually the nucleus is the same size as a red blood cell. It also occupies about 90% of the cell. The chromatin is deeply condensed and stains a deep purple, and here you will not see any nucleoli. The cytoplasm in these cells are small with a sky blue color. Here you may see very fine eosinophilic granules and vacuoles may be seen. You may also see them in movement or modal, so they might have their cytoplasm a little bit shaped like a mirror. The larger lymphocyte is a normal lymphocyte in development. It is 11 to 16 microns in diameter. The nucleus is slightly larger than the small ones that you have just seen. The chromatin can be more dispersed than the small lymphocyte. The cytoplasm has a lighter blue with peripheral basophilia around the edge, and here eosinophilic granules may be present. Now, if there's prominent larger granules seen, this is into the category of a large granular lymphocyte. These are a mixed population of natural killer cells and activated cytotoxic T cells. The nucleus is still round, So that is the characteristics that is not going to change with any lymphocyte. It can be indented due to its progression, but it's still round in shape. Cytoplasm is abundant here and very pale blue, containing coarse pink granules. Now, sometimes you may see a lymphocyte in the reactive stage. This is after the immunoblast transformation where they're activated in their presentation, and you might get a snapshot of what they look like. Activated lymphocytes or reactive lymphocytes, the cell starts to enlarge, okay? You may see dispersion of the chromatin, which is increasing in DNA synthesis and mitosis. So you start to see the cell growing in size, 16 to 30 microns, okay? And you'll see the nucleus is round or elongated or stretched. So we're still taking on the shape of a round to stretched type of nucleus. The chromatin is more dispersed here, and you may or may not see a nucleoli. The cytoplasm is increasing, and you're going to see basophilia around the edges. They also may scallop around red blood cells. They also can be called stimulated, atypical, transformed, variant, or activated lymphocytes. You can see very few in healthy individuals, but as soon as viral infections come into play, you may see more within the circulation. Now, an immunoblast is the next stage in blast transformation. They are very large cells, 12 to 25 microns in size. The nucleus is large, centrally placed. You may see a prominent nucleoli here. Fine chromatin, and it stains purple-blue. The cytoplasm is abundant and has a very intense blue color. Now, this cell will proliferate because of the producing antigen-specific daughter cells that will mature into either a plasma cell or an activated T lymphocyte. Now, plasma cells are identified in a separate category where you see a round or slightly oval nucleus. The cell is 14 to 20 microns in size, where the nucleus is eccentrically placed or placed to one side, and it has a coarse block-like radial mass of chromatin, where it can look like a spoke of a wheel or some kind of cartwheel. You won't see a nucleoli here, but the Golgi apparatus is obvious by the clearing zone around the nucleus into the cytoplasm. And it has a stretched out appearance. They have very high RNA content, and the cytoplasm enlarges due to the production of large amounts of immunoglobulin. You may have small eosinophilic granules present. Table 8.2 is a very good chart to study from about lymphocyte maturation because it describes the level of maturation, the shape, and the nucleus to cytoplasm characteristics. So first, as it's maturing, you have a lymphoblast. Next stage of maturation is a prolymphocyte. Then we get into the various types of normal lymphocytes. You have a small lymphocyte and a large lymphocyte. So look at the characteristics from the small to large and see that you still have one circular nucleus that is enlarging with the scant cytoplasm that was blue to a larger cytoplasm that is also taking up a blue color, but you may or may not start to see some eosinophilic granules here. The reactive lymph is a larger, stretched out, activated or changing cell. This is due to antigen stimulation, similarly to the immunoblast. This is where a foreign invader has been encountered and they start to transform into these activated cells. So please take a look at the characteristics between all of these types of lymphocytes. As well as a plasma cell, or a plasmacytoid lymphocyte, where you have the nucleus to cytoplasm characteristics. And these cells are B cells that are going to make immunoglobulin. So in the peripheral blood, the concentration of a normal lymphocyte count would be 1 to 4.8 times 10 to the 9 per liter. This is an absolute count. At birth, the mean lymphocyte count can be higher, and it rises to about 7 within the next 6 months and gradually decrease from the ages of 4 years old to adult. At this time, the infant is starting to build an immunity, and the conversion to lymphocytes happens. A couple terms here: lymphocytosis is an increased absolute number of lymphocytes of over 4.8 times 10 to the 9 per liter, and lymphocytopenia is a decrease in the absolute number of lymphocytes, less than 1 times 10 to the 9 per liter. You usually only see 5% of lymphocytes in the peripheral blood. 95 are stored in extravascular tissues such as lymph nodes and spleen. They move continuously between these intravascular and extravascular compartments, just waiting for foreign invaders. 80% of these lymphocytes are long-lived, meaning their lifespan is from months to many years. 20% of these lymphocytes are in the peripheral blood and can live from a few hours to a few days. Talk specifically about lymphocyte function. First up, B lymphocytes, and they are responsible for our humoral immunity. So as these naive B cells encounter an antigen, in the secondary lymph tissues, they proliferate and produce many B cells that can recognize that particular antigen if it's presented again. They differentiate into these activated B cells, or called memory cells, and these antibody-secreting cells are called plasma cells. They have a primary antibody response where the antigen binding to and activation of the naive B cells is associated with the production of antibodies. They have a secondary antibody response due to antibody activation of these memory cells, and here they develop more rapidly than the primary response because it's associated with IgG antibodies. They function to defend against bacteria, fungi, microbes. Antibodies neutralize and eliminate these microbes to their toxins, and antibodies produced by plasma cells in these lymphoid organs and the bone marrow circulate within the body. Plasma cell development are fully activated terminally form of B cells, and their primary function is synthesis and secretion of immunoglobulin that recognizes a specific antigen. T lymphocytes are responsible for our cell-mediated immunity. They protect us against intracellular pathogens. They secrete cytokines. They help activate B lymphocytes and macrophages. They recognize short peptide fragments from protein antigens bound to the MHC molecules on the cell surface. Natural killer cells are effective cells of the innate immunity. They function to kill infected cells and activated macrophages to destroy phagocytized microbes. They have direct cytotoxicity for cells infected with viruses, intracellular microbes, and certain tumor cells. They can also produce cytokines to stimulate and inhibit hematopoiesis. They activate cells of the innate and adaptive immune systems. As lymphocytes age, they decrease in immunocompetence over time. Similarly to the thymus starts to atrophy as individuals age. So these naive T cells don't respond as quickly or efficiently as they used to in older individuals. In result, the immune system is less able to recognize or distinguish self from non-self, which results in increasing autoantibody production.